No. It's okay. So first of all, I want to thank for the organizers and all people involved in this meeting. It's been a wonderful meeting. It's well set talks, and really enjoyed. I learned a lot. And I know it's really difficult lots of work to organize a symposium, and you guys really nailed it. It's congratulations. So I want to talk about some part of my project in the lab. So I'm interested in protein evolutions, any aspect of protein evolutions. Particularly, I try to understand the key elements to driving evolutions or restricting evolutions, and it's why some dynamics we understand if the general rule exists, and we can probably use it for the protein design and engineering. Obviously, lots of computational tools and AIs comes in evolutionary fields, and then I'm not sure I can survive in the next 5 years, but so we will see. So, but that's what I want to do. So that we do lots of different tools in the lab. I used to start it from the directed evolution tools to generate the laboratory evolution in the lab and try to see the pathways. And we get into the lots of protein bioinformatics these days and the combinatorial mutation analysis. We love to do lots of crystal structures analysis and the kinetics. Try to see from the molecular side to the evolution dynamics. I really want to go to the higher organism level as well. So we do the ancestor reconstruction. On some part, I'm pretty newbie, so I'm a little bit nervous about talking about it in front of you guys. But as we started to do the DMS, and we tried to expand a little bit more using those to study the evolutions. So the main topic I'm kind of getting into, if you want to talk about evolutions, this epistasis coming in always a topic. So that epistasis is just why most of the people understand this topic. And if you have the double mutations, and then how this mutation behaves, that's kind of decide how evolution behaves, essentially. So if you have two mutations, there's no interactions. It's called additive effect. There's nothing. 2-fold, 3-fold improvement in functions, combined 6-fold. That's kind of like log scale additive effect. That happens. But then it's interesting interaction happens. Some mutations synergize it, where you can do have two mutations, like 8-fold, 10-fold, 100-fold, that's synergize it. In some cases, it's kind of antagonistic interactions, and 2-fold, 3-fold improvement, combine it in the negative situations. In some cases, you have a positive, negative, combine it in positive as well. So they have a lot of those kind of stuff in evolution, and the prediction is going to be complicated. So that's obviously those right? How much we have, we don't know, but this dictates shape of the fitness landscape and how we want to navigate. At the same time, I want to understand how this interaction happening in the molecular level. So this is a very difficult thing. So I'm always struggling to understand it, but that's really ultimately we want to do. Okay, so the epistasis, we have to think about different level of the phenotypes, right? We can measure fitness or flux of the, if you're talking about the enzyme, I love about enzymes. So the flux or the fitness of the organism, but there's one layer of the phenotype you can measure it, and depend on the measurement you're doing it. And it's essentially down to the biophysical parameters. And the stability here, two-state folding is relatively simple problems. Everything is cooperating, folded, unfolded, if it's a two-state folding. And then you go to the functions, you have to talk about the limiting steps. And then all these reactions and happening in step system. So you may see some additive effect in those biophysical parameters, but when you look at the flux or the fitness, it could be more complicated behavior. So that's kind of, ideally we had to connect everything through the fitness effect. So the question is, why? Still I'm asking how much epistasis we have in the system. And then obviously, can we predict those kind of stuff, right? Well, lots of studies is being done, particularly from the Ben Leonard's lab here. Is lots of great work there. So what we see in usually, right, the epistasis is not really dominant if you look at double mutations and then all this combination. So it's kind of a simple world. But then the problem is if you have multiple mutations accumulated, and then let's say if you want to predict the fitness of the sequence in the 50% identity away, and it's become very problematic problems. Because it's a lot of small epistasis can change something is in the bigger in the long run, but also the higher order epistasis probably exists. It's not really big, but its accumulation of the mutation probably affect a lot. But when you look at the protein enzyme evolution particularly, if you collect all the mutations affecting function change, usually those are epistatics. So these are actually really epistatic, and then I will a little bit touch on this topic before going to the beta-lactamase. So when you have the problem is understanding epistasis and enzyme or the protein binding, it's always the complicated things, right? There's an interaction between, for example, substrate to the residues, and the residues to this interaction to the substrate as well. And it could be just cofactor moving a little bit. So it's obviously affected the catalysis. Some long-range interactions obviously happen, so there are lots of crystal structures and showing those kind of stuff. And the protein dynamics is obviously the key to understand this. And as well as some allostry is going on. So you have to really understand why the interaction of the residues to the functional state. So that's obviously way more complicated problem than predicting the structures. So this is one of the examples. I just wanted to quickly show this how messy it is. So this is the 5 mutation landscape. That's the evolution of organophosphate hydrolase. So this kind of historical 5 mutation happened probably in nature. So this happened for ancestral state, and then we identified 5 mutations to improve the 1,000-fold organophosphate hydrolysis. We saw the crystal structure, the 5 mutations is around, it's open up the active site. Then if you look at all the 5 mutation combination landscape, you see some epistasis here. So for example, this is kind of most likelihood path to go up, but you can see this mutation in 271, it's highly negative when you measure it in the wild type sequence, but you can see this actually path is going up toward to the end. So actually what's happening, 3 mutations has to come fast to flip this negative effect to the positive mutation. So some interaction happening to flip the effect of those mutations. So the complicated things you can measure, obviously different substrates. In this case, we can measure this is the thiol here, this is an oxon here, so the different the shape of the substrate, and then actually it's changed the fitness landscape here. Right? So you see this is a negative here, but then when you change the oxon, it's actually there's a very positive mutation at the beginning. So this actually clearly the substrate to the enzyme is interacting, but also the part of the substrate is actually clearly interacting to the certain residues as well. So you can map the certain epistasis effect, that's what we do. So there's kind of the active sites here. So you can see that actually the most prevalent interaction is you can see it's through the substrate, not between the residues. So you can see, but also the change of the substrate, some change the interaction. We don't see any particular interactions between the substrate and the enzyme in the crystal structures or the MDs and everything. So it's very complicated things, and we try to figure out a little bit more, doing more experiments. So this is kind of the overview of how adaptive landscape is complicated. We kind of did the same analysis in the different systems, and some of them are our systems, and the others published data. So when you look at the overall, this is actually when you see the distribution of single point mutational effect in the entire landscape, you can see over this kind of all these distributions. And some mutation actually change 1,000-fold difference in between different genetic backgrounds. When you look at a sign effect, more than half of the mutation actually change the sign, depends on the genetic background. So it's kind of positive to negative, negative to positive. And the rest of those also the change the magnitude as well. So it's like 70%, 80% of the mutation is epistatic. When you look at when you look at from the wild type, it doesn't actually match with average effect. So the wild type sequence you started to do the evolution, it's not really kind of representing the entire landscape. It's kind of the outline as well sometimes. So you can see it's kind of deviated like orders magnitude. So the epistasis kind of same things. This is kind of the epistasis factors. So there's like 2 pairwise interaction scores. So you can see actually the several orders magnitude, the difference in epistasis, and depends on the genetic backgrounds. And second order, third order, fourth order, you can see the same things. So those mutations obviously collectively changing something to substrate or the chemical reactions, and that's obviously prone to the very epistatic. So this, I just wanted to show this is kind of the different problem, maybe the different problem than looking at the protein folding. Okay, now I can start with the beta-lactamases. So that's why I've been working on this system. Partially tried to get the funding from the Medical Research Council, but also this kind of an interesting system because they have multiple evolution events happens. It's massive families because it's probably started to evolve in a billion years ago along with antibiotic producers. Then it's obviously a great protein evolution system is because selection is so easy to do. But I try to understand everything this enzyme family is going on. So lots of open questions. How much actually distribution of beta-lactamase in the environment, we don't know. Which extent, why phenotypes are different, and the level of that efficiencies and substrate specificities, we don't know much about it. Also, what's the difference between the beta-lactams came into the pathogens or stayed into the environmental microbes. And we want to see like why some difference in evolvabilities. And eventually we want to do the sort of models to predict sequence to the phenotype to the substrate specificities. Okay, first of all, we need to understand why the distribution of those sequences. So what we do is kind of comprehensive bioinformatic approach. We take every sequence from the NCBI and take the entire superfamily level, not family level, the people classified. And I'm going to do sequence similarity network. So this is kind of metal beta-lactamase superfamily, but you can see these two clusters here are beta-lactamase. The rest of the sequence are most likely non-beta-lactamase, different hydrolytic enzymes. You can extract those sequence. Obviously, you can make the HMM, and then you can go to the metagenomic database, so they extract more sequence from the shotgun sequencing data, and then they remake the sequence similarity networks and then phylogenies and try to see what's really the cutoff between the beta-lactamase antibiotic resistance to the non-beta-lactamase resistance. So this is kind of the overview of the phylogeny. So this is two superfamily exists. So one is the, this is the PBP, so the penicillin binding protein superfamily. And then there's three independent evolution happening, class A, class C, class D here. And this is a metallo-beta-lactamase superfamily, and then two independent evolutions seem to happen. So there's actually 5 independent evolutions happen to degrade in this beta-lactam. So we had to do everything for the bioinformatics. So we collect all the sequences in NCBI. So it's where you compare to what the current antibiotic database is showing is like about thousands of sequences, but you can extract actually about 10 or 100,000 sequences in those databases. So they are coming from the environmental microbes, and then some of those is transferring to the pathogens in every year, and then people talk about all the super bags and stuff. So this is kind of the one of the figure for the class B enzymes. So you can see these are big clusters. Most likely everything is beta-lactamase, a little bit connected to other clusters. This is class B, and other family exists, but the other sequence most likely not beta-lactamases. And you can obviously draw the phylogenetic tree and then map all this the pathogenic related to the sequence. You can see some part is actually not really transferred to the pathogens, but it's actually beta-lactamase as well. So we can do the different classes. You can see this, there's the different analysis here. So then you can really extract every sequence, most likely the beta-lactamase. So I just wanted to show another snapshot. That's what we can do. You can actually, because you're collecting all the NCBI database, also the JGI metagenomic database, and you can actually trace each classes, which taxonomic distribution of the host, also which environment usually those samples being taken. For example, this one is contained NDM. It's kind of, now it's most famous, the class B beta-lactamase is mostly coming from this class, group of sequence coming from aquatic environment and from gamma proteobacteria. So if you go dive into it, most likely we can trace actually the origin of the host that's why transferred to the pathogen on those days. So you can do those kind of stuff as well. All right. So going to the epistasis now. So that's the predicting what this different sequences, difficult things, and then we want to try to see how much this epistasis exists. So that's when we started to do the DMS. So this is our approach. Make the library of the DMS library, a single point mutation library, and then go for the different concentration of antibiotics on 8 or 10 different concentrations. And then one side we can measure obviously the fitness score, different compared to the wild type sequence, but also we can do the dose response curve of each variant, so that you can draw it and extract the EC50. So you can do the two different measurements here, and then EC50 nicely correlated with a single isolate as well. So we extracted a few different parameters. So this is kind of a nice the data set to consistent. This system is working really well. So the EC50 that's we extracted from the dose response curve. And then we can also compare to the fitness score that's in individual concentrations. And then you can see the nice sigmoidal curve in every concentration. So that's actually telling us is there's no hitchhiking going on, and it's each individual variants responding to the antibiotics. That depends on their EC50s. So because of these simple figures, and then we can draw every single data set in one map, EC50 here, and concentration of antibiotics in the other side, and the fitness score in the axis, and you can fit it in one equations, and it's pretty simple beta-lactamase fitness landscape. Obviously, it's based on our condition of the experimental conditions. But this behavior of the bacteria against the beta-lactam is very simple. Okay, so we can do this kind of stuff. And now we try to see the epistasis and doing the DMS in two different enzymes. So BIM and NDM1. So the two enzymes is about 30% sequence identity, so 70% of the positions are different amino acid. When you look at the phylogeny, ND1 located here, BIM2 is located here, so it's kind of actually the neighbor in terms of the clinical pathogen related beta-lactamases, but it's still the very, very distant sequences. So that obviously so we want to see how the two DMS is showing us different effect. Also, we want to see how the model can predict these differences. So we are collaborating with Martin White in Sorbonne University in Paris. So they started to do the DCA analysis. So essentially, they look at the alignment and then looking for some conservations, as well as the residues on the position is co-varying between the different positions. So this is kind of the base on the AlphaFold prediction models. And then they can do sequence generation and then looking at all the sequence kind of value estimations, and then they can do the fitness prediction. So that's kind of the simple model, simple biophysical model to explain fitness of the proteins, and then we want to ask how much this system can explain what we see. So this is sort of DC analysis. So this is each different sequence of the beta-lactamases, and then the predicted mutational tolerance. So when you look at 19 different amino acids, how they are tolerated. So it's kind of average effect. So it's a bit difficult to do everything. It's in one figure. So it's just average effect for the 90 amino acids. On the red one, it's really conserved. They don't like to change it. So this obviously the conserved residues. And then depending on the sequence, certain intermediate sequences has varied effect. Okay, so that's the model predicting this way. And then we do the 2DMS, and then it's compared to those effects. And this is kind of the same, sort of same analysis. So we look at the tolerance of each positions, 90 amino acid average, and then two different DMS data sets you can see. So it's, some are obviously, it's the very deleterious one is kind of clustered in one positions, but you can see there's a scattered dots in both sides. So it's why the 2DMS doesn't agree perfectly in here. So we compare DMS to the DC analysis. So you can see some correlation is there. So again, it's why the very deleterious mutation is predicted in both ways. And then maybe the non-predicted ones are non-deleterious one is okay. But when you can say this is okay, nice fit. But if I remove the both extreme side, it just is, for me, it's random scattered dots. So this actually the problem exists in the predictions. Why this you can get like R squared 0.5, it's not terribly bad. But then the reality, this is the kind of the mess, and this is actually the mess we are having a problem to predict the sequence and functions. So what's the cause of these kind of the weak correlation in the middle part? That's the questions. Now we look at the different ways, and we ended up only the very simple way. This is just surface accessibility of the residues. This is kind of the easiest way to explain. So this is the two equivalent figures and DMS from DMS and the DCA. So the two enzymes scores kind of averaged, and then it's plotted against the ASA. So this higher one is the residues at the surface, and the lower numbers is the residues at the core. Right? You can see this way, the nice agreement is the both sides. So the surface residues are highly tolerated, and then this core residues are very difficult to predict. But then this middle part is sort of second core of the protein folding, and this is the where mess happens. So we try to do the residual level comparisons, but we did the average one. So it's a bit complicated to compare. So there are certain positions shared, exactly same amino acid between two. Certain positions of different amino acids, so we classify it into different ways. But then we have the two different amino acids, but if the DMS showing this NDM to the BIM mutation, BIM mutations are neutral, we just treat it as the same amino acid, okay? So in this case, we can compare the 90 different amino acids together. But this case, we just only compare mutation reversion between NDM to BIM and the BIM to the NDM, okay? So the first one, this is kind of the 90 different amino acids, and then, right, about 130 positions. So you can see again, right, so the neutral mutation is easy to predict. Dead one is easy to predict, but you can see lots of epistasis in between. So those one we mapped onto the structure. Again, you can see ASAD predict nicely. So the surface is where the both sides is low epistasis we can observe, and the core is again the low epistasis we can see, but the middle part is exactly the problem we have. The same things in diversion. So we're just looking at the two, the diversion in between. So you can see some mutations agree a lot, but there's fraction of the mutation that they don't like each other. So this kind of compatible one is like this agree with it. And then the two categories we decided. So both wild type sequences kind of the negative in both sides, kind of sign epistasis going on. But the other side is one is tranched, the one is negative, one is neutral, but you can see the epistasis effect. So how much the DCA, right, predicted those kind of effect. And you can see the compatible one is relatively okay to predict. But the problem, again, starts, and particularly this double tranched mutations, both negative to exchange, is very difficult to predict from the DCA. And those ones is particularly on this kind of middle surface residues. All right, so that's kind of what we see in the 2 DMS. And what's the next? So we try to expand this, and then we are challenging our collaborator to predict more. So we're kind of playing with the massive gene synthesis platform. So this is called the DropSynth, that developed by the Katie Press and University of Oregon. So we can synthesize thousands of the genes in-house. And then obviously we can do a lot of phenotyping, and whatever that they tell us what the sequence is going to be active or not, and then we can try to test it and then feedback it, and hopefully we can do some kind of better modeling. So if you're interested in, I'm happy to collaborate with any people to do this system. All right, what else? So I want to show you another example that we explore in the fitness landscape. So analyzing those extant enzyme, different sequences, it's nice. We can see lots of things, but we're missing all intermediate of the evolutions. So I want to see the step-by-step change in how these epistases accumulate in the system. So the only way we can do is laboratory evolutions, but it's a really extensive way. So that's why we can do the mutagenesis and the plating and antibiotics, and then just scrape at those guys, and this is one round of experiments. Obviously, you can measure the sequence and then the variants, and then we want to do the very extensive way. So that's kind of long-term project. Maybe we started like 10 years ago. So took the wartime beam and tried to evolve as much as high antibiotic resistance, so that we changing the antibiotics every round, tried to push as much as high. Then we really get to the seeding. Almost we had to put the entire bottle of the antibiotics into the solution. Then we switched to the different phase, and then two different stage antibiotic concentrations, and then just stay this concentration almost 100 rounds of evolutions. Okay, they're just repeating this. It took us about 2 years to do it. Poor students have to do everyday transformations. So what do you expect in the fit? The resistance, it just goes up, obviously. And then when you switch to the neutral drift, right, this constant antibiotics, high concentration, you expect to see a high resistance level, and when you lower it, you expect to see going down and then staying here, okay? And then you mutation accumulate whatever the way they like. All right, so this is kind of the mutational accumulations. So you see this is the nucleotide mutations. We accumulate about 150 mutations out of 800 base pairs. So still 25% change, so not terribly big, but this is actually good enough to see something. So amino acid level is about 60 amino acid mutations in low antibiotic concentrations, and you can see this sort of pseudo DM DNDS ratio, so that at the beginning of the adaptive evolutions is the amino acid mutation accumulate more than nucleotide mutations, so they try to change the phenotype. Then after switch to the sort of neutral drift, DNDS drops it, and the nucleotide mutation more accumulate because there's a sort of purifying selections going on. So it's kind of nice population genetics going on. I have to say there's lots of recombination happens because we use a PCL. So it's kind of promiscuous heterosexual system, not asexual system. So the different phase of the evolution is being done, adaptive evolution, so kind of relaxing the purifying selections, while dropping the antibiotic resistance, antibiotic concentrations, and then two phase of the threshold in a neutral drift. So we can draw, obviously, phylogeny. We didn't do the high throughput sequence yet, so there's the Sanger sequence and everything, like 500 variants or something. So the T-thousands, high concentration of the lines here, and then lower concentration of the lines goes there. So it's thankfully there's not much contamination, cross contamination in between. We are working on trying to understand these sequences. Lots of APCs going on. Some fixation happens in some of the lines. But I can't show you because it's still too much mess. So the next time we can probably show you. But there's something interesting we can see in phenotype level, telling us some important dynamics in evolutions. So this is kind of the fitness resistance level of the populations. So when we measure the resistance level of the population, there's a kind of war type, and it's obviously going up here, and switch the neutral drift, and high concentration of antibiotics stay there, and the low concentration you can see drop and stay here. Well, nothing interesting, maybe you can say. But you can see this kind of error burst here. We call the phenotypic diversity. It's not like an error rate in experiments. What we see is actually different shape of the dose response curve. So the gray one here, maybe it's difficult to see. The very steep one here is war type, the BIM. So when we have the single sequence in the population, you can always see that this is a steep curve. And then after the adaptive evolution, you can see the very steep curve here. The resistance level up about 1,000-fold, or maybe 100-fold, and then still very steep. So that's telling us that why phenotypic variation in the population is very uniform. And then after that, this neutral drift in this low concentration actually started to drop it. You can see the median is going to drop it, but it's actually the curvature, it's not getting steeper. So you can see this is actually is the distribution we see in here. So we isolated the individual variants, determined the mix of each variant. So you can see this is a kind of selection concentration we use, but you can see this massive distribution within the populations. So there's some variants actually 200-fold higher minimum inhibitory concentration than we use in experiments. But these guys are going through the 100 rounds of the evolutions, accumulated more than 60 mutations, but still, actually, it's not much drop from what we did adaptive evolution. So that's kind of interesting is going on. So what's happened here, it's mostly my speculation here, but there's a simple dynamics probably going on. So when you look at this fitness distribution or the EC50 distribution here, you can see this is the wild type populations, and then you can see some of the mutation to increase the resistance. So when you do the adaptive evolution, obviously those ones being picked, going up, 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 and then get to the seeding. But then when you do the nutrient drift, well, most of the mutations are deleterious, obviously. Those are purged out. The neutral mutation accumulate and stay there. But these are the almost right top of the seeding, and they just stay there. But when you have low concentrations, well, everyone can go down, but at some point, actually new adaptive mutation comes in. Because you see the why I showed this fitness landscape, actually top is pretty much plateau. Where they don't care higher resistance. It's the same fitness, but they can be higher resistance, no problem. So I suppose you have the mutation to increase resistance level, and those ones comes in all the time. It's like kind of equilibrium between positive mutations, and then all the negative mutations will be purged out anyway. So we are kind of playing with positive mutations and the neutral mutations. So then, as you can see, end up with a really huge diversity of phenotype without any selection happens. So we try to actually oscillate the antibody concentration, but it's just a one round we can get it back to these evolutions. So that's most likely happening. When we do actually the selection without lower than E. coli resistance level, and every enzyme is going to die. Stop codon comes in, and it's every enzyme is dead enzyme after 5 rounds. So you see this kind of the evidence in here. So when you see that this tree is actually the dim of the color is representing antibiotic resistance level. You can see those kind of high resistance guides is appearing in the middle. It's not like one cluster staying, not getting any deleterious mutation or anything. It's kind of the exchange happening all the time. All right, so that's the summary. So I showed the different aspects. So I'm over the time, and I just wanted to acknowledge the people who worked on this, and thank you for your attention.